All right, in part two here, we're going to start talking about the different phases of mitosis. And what we can do is we can look at them as two kind of like larger phases in that we have interphase and then the smaller phase is mitosis. But collectively, they make up really the cell cycle. And then within interphase, what we can do is we can break it down into G1S and G2. And then mitosis can be broken down into prophase, prometaphase, metaphase, anaphase, telophase, and then cytokinesis. And we're going to go through each of these here in the next few minutes, okay? So if you look here again, here's the entire cell cycle itself. And you can see interphase is going to occupy the vast majority of time. Mitosis is only a very small amount. So when we look at G1, S, and G2 here, you can see we've got G1 right here. Change my color back to black here. So we've got G1, and then we've got the S phase, and then we've got G2. So that comprises interphase, okay? And then if you look at mitosis, you've got a whole host of different types of things with mitosis, and really that's what's going to be taking up the M phase right here, where we're going to kind of divide this cell up and then split the cytoplasm and end up with two new cells, okay? So interphase now, during interphase, the G1, this is, it should say a lot of different types of genes here are turned on, okay? And this is what's going to be really making the proteins that are necessary to run the metabolism of the cell, enable the cell to do its job, and this make, you know, some of the examples here that we've got, you know, the expression of the genes that make the enzymes, break down carbohydrates, lipids, proteins, etc., just the thing that the cell needs. And then during this time also the cell might be getting larger, okay? And as it's getting larger, of course, recall what we talked about early on in the year with the whole surface area to volume ratio, that's becoming a lot smaller, and as a result, you know, some of the mechanisms are in place. It kind of senses this. The cell doesn't have the ability to kind of get the things that it needs or get rid of the waste that it needs, so that might be what's going to initiate the cell division, okay? And then during interphase also, we have that S phase that we talked about. And just think DNA synthesis. This is where the DNA is going to be synthesized or made. And, you know, in that process, we're basically duplicating the amount of DNA. And we're going to, you know, have a variety of checks in place to make sure that this process is occurring correctly, okay? So with a G2 now, this is where the cell goes G1, then S, now G2. This is where the cell is going to prepare to actually divide. And there's a lot of different proteins that are starting to get manufactured, a lot of things that are coming into play, these little kind of protein ropes or these microtubules, these spindle fibers, so that the genetic material can get pulled apart. Organelles are going to grow, divide, increase in number, etc. And what we want to make sure is that each cell contains what it needs in order to support itself and survive, just like that original cell did. And once you get towards the ends of G2 now, things start to slow down as this cell starts to ready itself for division. So the first part of mitosis here, the M phase, is prophase. And this is where you can start to see the actual genetic material. So if you're looking right in there, you can see it's starting to condense down, starting to become highly coiled like we talked about in the last part. Prometaphase now, the nuclear envelope itself is starting to break down because we have to allow these microtubules access to the kinetochore, the centromeres of these chromosomes, so we can start to pull them apart. So during prophase now, or prometaphase now, you get further condensation of the chromosomes and the breakdown of the nuclear envelope. With the M phase here, this is characteristic. When you look right here, you can see these chromosomes are aligned at the metaphase plate. Okay, so that arrow is pointing really at the metaphase plate. This is where the chromosomes are aligned, and you know, any cell that you look at that looks like this is in metaphase. I mean, it's just very characteristic of it. The other two here, prophase and prometaphase, are a little bit tougher, but with metaphase, this is obvious. Okay. Now with anaphase, what happens is we talked about the sister chromatids being joined at the centromere and all along their lengths in part one with these different proteins. And during the process of anaphase, those proteins that are holding those sister chromatids together start to break down, start to become inactivated. And what happens is this allows, when these microtubules are attached, when they start to kind of pull apart, this is what it's going to allow those sister chromatids really to separate themselves. And then you'll have some motor proteins that are going to be pulling the two sister chromatids to the opposite ends of the cell, so that when the cell actually goes through cytokinesis and divides, the cell has everything it needs in order to kind of function like that original parental cell did. Okay, so if we're looking at anaphase now, this is another characteristic phase. You can see that these chromosomes that are aligned at the metaphase plate kind of begin to pull apart here. So you can see they almost look like, fingers kind of sticking out, kind of pointing in this direction and pointing in this direction here. But this, this is characteristic of anaphase. These chromosomes are starting to move apart. Now with telophase, this is a little bit more difficult to look at. It might be easier when we do the onion lab to kind of see these types of things, because these look in some respects very similar to prophase or prometaphase. But in telophase, what happens is you start to get these daughter nuclei reforming. The genetic material gonna start to relax back down, and you've got this obvious kind of delineation here between this cell and this cell. So you're getting these new nuclei and two new cells that are actually starting to form. And then during cytokinesis now, you're starting to pinch the cell in two. All right, and again, when we do the onion lab, we're going to see this. In an animal cell, we're pinching it in two. In a plant cell, we're kind of building the cell wall between the two nuclei of the cell. Okay, so in animal cells now, there's a difference between the animal cells and the plant cells, and you're going to want to make sure that you know the difference here. And you get this cleavage furrow in an animal cell that starts to form. And what happens is it's kind of a contractile ring that's going to pinch that cell in two and basically form two new cells. Okay, and what you can see is that you have these actin microfilaments really that are going to interact with these myosin molecules kind of on the inside in the cytoplasm of the cell, and that's what's going to kind of pinch this cell in two. So if you look at this electron micrograph right here, you can see the contractile ring that's kind of pinching, forming that cleavage furrow. And the way you can kind of think about it is if you have art class and you work with clay and you've got the wire that you can kind of cut the clay apart with, that's kind of what it's like. So you can pinch these things in two. And this contractile ring, again, takes this initial kind of single cell with two nuclei, pinches it in half so that you end up with two daughter cells that are genetically identical to one another. Okay, plant cells, it's a little bit different. Okay, because what you're going to do is you're not going to form any sort of a cleavage furrow. There's no contractile ring or anything like that. What you're getting is these vesicles. And if you recall what we talked about early on in the year, how we make proteins on the ribosomes. If the ribosomes attach to the endoplasmic reticulum forming the rough ER, those proteins that get made into the lumen of the rough ER migrate to the Golgi, where they're going to be modified and then exported from the cell. So those bound proteins are going to be bound to the ER. They're going to make proteins that are destined outside of the cell. Well, here's an example of that. So what happens with plants is you get these vesicles, these proteins and such that are made along that pathway. They migrate to the Golgi, and then the Golgi kind of gives off these vesicles that are going to attach to the microtubules and get kind of exported towards the center of this cell. And what you're going to do is you're going to lay down the cell plate, which is eventually going to form the cell wall. So these vesicles are all going to be kind of interacting with one another, and you're going to get two new cells formed. So if you look here, you can see these are the vesicles that are kind of migrating towards the center here, and there's a bunch of little ones here and so forth. And once you look at those things, you can see that the same thing is occurring on this side. You've got a bunch of vesicles and so forth, and they're all migrating in that direction. And then basically, here's an example of the vesicles in one, and then as they start to fuse together, they start to form this cell plate in two, and then eventually this new cell wall gets formed here in part 3, and then now you've got two daughter cells from this original cell that got too big to kind of handle the surface area to volume ratio and that sort of a thing. Okay, so that's the difference between plants and animals. Plants form these cell plates and cell walls and kind of divide that way as the cell elongates, and the animal cells have this contractile ring that just pinches the cell in two. Okay, so here's a little bit of a breakdown when you want to look at this. You can kind of pause your video there and zoom in on it and kind of look at and just notice some of the differences between mitosis and the different phases of mitosis, and this of course would be in plant cells. Okay, and you can just take a look at that. If you have any questions, of course, write them down. Now, centrosomes, these are non-membranous types of organelles that you find in animal cells. And sometimes these are referred to as the microtubule organizing center. And what they're going to do is give rise to the microtubules that are going to extend out during interphase. They duplicate and eventually migrate towards the opposite ends of the cell. And then what they're going to do is really, like if you have this cell here, they're going to migrate towards the opposite ends of the cell, and then you're going to kind of attach in a way to the cell membrane here, and then here as well. And then these microtubules kind of extend out, and what they're going to do is they're going to join up with the chromosomes here at the center and kind of pull, as they pull apart and separate, they're going to pull those sister chromatids towards the opposite ends of the cell, so that when that cell pinches in two, what you end up with is two cells that each contain the appropriate amount of genetic material. Okay, and the microtubules, we said, grow out from them. They attach to the kinetochore within the centromere of each of those sister chromatids, and again, just kind of pull it apart like I described to you a second ago. And what happens initially, though, is we said that all of those sister chromatids are kind of joined all along their length by these proteins. And then once those microtubules kind of attach to the kinetochore, you kind of get this tug of war taking place. And that's what's going to align those cells really at that metaphase plate. And then as these proteins along their length begin to break down, that's when these cells start to kind of pull apart and divide during anaphase. Okay, and that mitotic spindle is really important, because this is the way that the cell can kind of move these chromosomes around during the cell. So you can see all of these processes that are taking place. We're condensing the chromosomes, we're making the DNA more manageable, we're duplicating it, and we're putting it in a way when we organize it that we can kind of grab a hold and pull these things apart to ensure that both cells contain the appropriate amount of DNA. Okay, so you can kind of look here and see some of the things. Again, you can pause the video and see some of the things that we've talked about here. And when you're looking at it, again, you can see the centrosomes are kind of joined and attached along the surfaces of the cell. And you can see here's the metaphase plate. That's where that tug of war is taking place. And then during anaphase, these things start to pull these sister chromatids towards the opposite ends of the cell. And then during telophase and cytokinesis, we're going to end up with these two daughter cells here at the very end. Okay, there's some, just, I guess, color being added to these things, and you can see kind of what's going on in terms of the condensation of the chromosomes, the spindle fibers, they're in kind of the greenish yellow color being formed, and metaphase, anaphase, telophase pulling these things apart and so forth. Okay. So now the next part here, part 3, we're going to be talking about the different stages of the cell cycle and kind of how we know these events are occurring. All right. So any questions you got, make sure you write them down, and we'll talk to you soon.